Sorry. Hi, good afternoon to all of you. Let us continue the topic that we discussed in the morning section. It's nothing but the experiment conducted by the group. And this experiment to prove that the real genetic material is DNA, not RNA or not the proteins. So once again, just to go through the experiment conducted by the group to get the continuation. He has selected two type of pneumococcus bacteria, and one is what the rough non-virulent, another one is smooth virulent, and rough non-virulent injected in the body of the mouse, and this mouse it's healthy only after injection, whereas smooth one is what the strain is virulent. So naturally, what happened? This rat died, mouse died due to the pneumonia, and injected that heat killed a smooth virulent strain, but their mouse lived naturally because it is what the dead heat killed only. And he mixed the rough and what you can say heat killed a smooth virulent and injected in the body, and he found this mouse dies. And he collected this blood from this mouse and find these living strain bacteria in the blood sample. Actually, he injected only that heat killed what you can say smooth strain here, but in the blood he found this what you can say the blood from the blood sample of the dead mouse. He found the living as smooth bacteria. So this is what the thing we discussed in the morning. So let us see the conclusion of his experiment. See, Griffith concluded after this experiment that the R strain, it is non-virulent. This bacteria somehow been transformed into a strain because he injected only the dead body of the strain, heat killed strain only. But somehow that R strain, it is a living one, transformed into a strain, and by the heat killed a strain bacteria. And he called it as what the transforming principle, because here that R strain bacteria is transformed into S strain bacteria after mixing. And he called it as what the transforming principle only. The reason for the transforming of non-virulent R strain into a virulent S strain due to the transfer of some material from the heat killed S strain to that R strain only. Whenever it's mixed, you know that S strain is heat killed only, but some material present this the heat killed S strain, it transferred to the R strain, non-virulent R strain, that only convert the R strain into what the S strain, that only transform the R strain into S strain. And this transferred material, he call it as what the genetic material. The material which is transferred from the heat killed S strain to the R strain and help them to transform from the R strain to S strain is called as what the genetic material. And but he failed to explain the biochemical nature of the material. He was not knowing whether it is protein, RNA, or DNA. He failed to explain the chemical nature of the material which is transferred from the heat killed strain to the R strain, make this R strain to transform into what a virulent strain. Then come to the biochemical characterization of this transforming principle. And before this experiment, this Oswald Harvey, then Colin MacLeod and McCarthy thought that this genetic material is a protein only, it's to be a protein. They worked to determine the biochemical nature of the transforming principle in Griffith experiment. And they purified the biochemical, like a protein, RNA, and DNA from the heat-killed S strain bacteria to see which one is converting the non-virulent R strain into the virulent R strain. The Griffith told that something is transferred from the heat-killed S strain to the R strain to transform it. And they tried to purify this biochemical present in the heat-killed bacteria, that S strain only, and identified which one is really transformed the R strain into the S strain. They have isolated the protein, RNA, DNA to find out which one is responsible for this transformation. And they discovered that DNA only transforms the non-virulent R strain into a virulent S strain. They have tested with these three biochemical purified from the heat kill strain, protein, RNA, and DNA. With that, they tried and find out finally DNA only transform that non-virulent R strain into what the virulent S strain. That only caused pneumonia in this mouse, and finally that mouse died. So they also discovered that protein digestive enzyme, that is what the protease is, and RNA digestive enzyme, that is RNases, did not affect the transformation. Actually, after isolating these three type of biochemical from this, what you can say, heat-killed S strain, what you can say, mice, after injecting the mixture of heat-killed S strain and R strain, they applied the proteases to destroy all the protein, and also applied these RNAs to digest all the RNA. And he did not affect what the transformation. It did not. After the destruction of the protein or this RNA, they found what the transformation. But the transformation substance was not protein or the RNA, but the digestion of DNA with the DNases did inhibit the transformation. So then they destroyed the DNA. They destroyed the DNA and done the experiment and found that it inhibit the destruction of the DNA by using the DNases. They inhibit the process of transpiration, and it is proved that DNA causes the transformation. DNA only causes the transformation, not the RNA or not the proteins. And they conclude that this DNA is what the real genetic material, but all the biologists were not properly convinced. They have explained the chemical nature of the genetic material in this transforming principle with all these examples, but this, what you can say, the biologist of that period was not properly convinced. So what is the difference between DNA and DNases? This question asked in the textbook, DNA and DNases. I told you DNA is what the genetic material, but DNases, it is what the enzyme to digest the DNA. Similarly, proteases is the enzyme to digest protein. RNases are the enzyme digest RNA. So destruction of RNA or protein never affect this transformation of this what you can say, R strain into S strain. But whenever they destroy the DNA present in this material, it inhibits the transformation. That means the DNA only responsible for converting this R strain into what the virulent S strain. So this is what the biochemical explanation for the transforming principle. Then another experiment conducted by the Hershey and Chase. And to prove that DNA is the real genetic material is DNA, it's not proteins. This is what the second experiment. To prove that DNA is what the real genetic material, not the RNA. And this experiment conducted by the Hershey and Chase. So we call it as what? The Hershey and Chase experiment. They conducted the experiment in what you can say, the bacteriophage only, or the phage virus only. So here, the Alfred Hershey and Martha Chase in 1952, they worked with a virus called bacteriophage or the phage virus. You may be heard about this virus, bacteriophage or a phage virus. It is what a special type of what the virus only, it is attacking only the bacteria to multiply its number. And this bacteria is having a genetic, this Virus is having a genetic material that is DNA, double-stranded DNA only, and it is covered with a protein coat. All of you know the structure of virus. It is not having any cellular structure, and it's made with only a pair of DNA in case of what this phage virus or what you can say bacteriophage. And for the protection, it is having a protein cover over it. And this bacteriophage consists of double-stranded DNA, and it is protected with a protein coat outside. And this virus infect bacteria and send its genetic material DNA inside the bacteria. Whenever this bacteria is infected with this virus is sending only what the genetic material inside, that DNA only it is sending inside, and protein part will be there on its outside. It is not taking the protein inside. Only it send the DNA. How it is multiplying? Its number in the bacteria. This DNA, after taken inside, this will go for replication many times and produce many copies. And this DNA individually produce what the protein by using the machinery available in the bacteria, because every virus required a host to multiply, because they don't have any cellular structure to make its copies. That is why they are always depending other individuals to multiply. Similarly, bacteriophage is also sending only its what the DNA inside the body of this bacteria. After sending this DNA into the cytoplasm of the bacteria, now it's go for replication and making many copies. And later, after making many copies of what this DNA through the duplication process or replication process, it is making a protective cover by utilizing all the machinery present in the bacteria. And after preparing many copy, naturally it break the wall of this bacteria and it comes out. And that is why we call it as what the bacteria killer. It killed the bacteria at the end. After multiplication, they want to come out. So it breaks the cell wall and it comes out. Right. Then outer protein coat of the virus will be outside only at the time of infection. The bacteria at the time of infection, it is not taking the protein inside. And the inserted DNA will multiply inside the bacteria through DNA duplication or replication and prepare new protein coat by using the bacterial machinery. And after multiplication, bacteriophage kill the bacteria and come out by bursting its cell wall. So this is what.
One second. I think
It's not going.
no, yeah, actually, it's going, but it is very bright.
Cannot reduce the size. Huh?
Is yeah, for you can reduce it. Check. It is in the PPT like this only.
No, no. So one.
Okay, okay, one second.
Like that only?
Is it like this, right?
Okay, then no problem.
It's like that. We'll continue, right?
Okay, sorry for the inconvenience because of some technical problems only it's interrupted in between. Then see that this is what the bacteriophage can find. This is what the bacteriophage or the phage virus, and it is infecting this bacteria. This is what the bacteria. It's infected with this bacteriophage, or it's called as what the phage virus. Why it is called as what the bacteriophage? Because it infect only the bacteria. What is the purpose of infecting this bacteriophage into the bacteria? It is only to multiply its number. As I told you, in case of virus, there is no machinery to multiply. It is having only a pair of what the DNA, and it is located in the head region. In the head region, only this DNA is located. And actually, at the time of infection, it is sending only the DNA inside the body of the bacteria. And inside the body of the bacteria, after sending this DNA, it go for replication and multiply its number many times. After replication, after getting the proper number of what this DNA inside this bacteria, by using that bacterial machinery, like what you can say ribosome, RNA, means what the tRNA, rRNA, mRNA, then amino acid. These are all used by this what the DNA sent by this bacteriophage inside the bacteria and prepare its protein. After preparing the protein by utilizing the entire machinery of this bacteriophage, it is making the protein code. And after making this protein code, it breaks the wall of the bacteria and all the bacteriophage will come out and destroy this bacteria. And that is why we call it as what the bacteria killer. See how this bacteriophage is infecting a bacteria. And this is what the bacteria, and this is bacteriophage. So bacteriophage infect this bacteria, and it is what the viral DNA or RNA. In case of what the bacteriophage, this viral is having the DNA only, double stranded DNA, and this bacteriophage, send the DNA content directly to the cytoplasm of this bacteria. After reaching this, what the viral genome inside the cell of the bacteria, this genome, viral genome like DNA, utilize the entire what you can say machinery present in this bacteria to multiply its number. It keep on multiplying many times. It multiply through the process of what the DNA replication, and after multiplying, what happened, this is what the infection first steps is what the infection, it is attaching this bacteriophage on the body of bacteria, and this is what the bacterial DNA, and red colored it is given in the head region of what this bacteriophage, that is what the viral genome. And during this infection, it penetrates that DNA content of the bacteriophage only penetrate and it is taking inside. And by using this DNA, it produces RNA, and by using all the machinery present in the bacteria, it prepare what the proteins. And many times the DNA will multiply. Every DNA after multiplication go for the what you can say RNA production and protein synthesis and making a protein code, and many such type of what the bacteriophage will be produced inside the bacteria. And after producing many number of bacteriophage inside the bacteria, it break the ball, kill the bacteria, and everything will come out. This is what the cycle of what this bacteriophage only. And this phage virus only utilized by the Hershey and Chase to conduct the experiment to prove that the real genetic material is DNA, not RNA or not proteins. So let us see the experiment conducted by the Hershey and Chase in detail. And they grow some virus on a medium containing radioactive phosphorus, that is P32, radioactive phosphorus P32, and some other medium containing radioactive sulfur, that is sulfur 35. They used the two medium. And in one medium, they cultivated some viruses, that is in P32 only, and some other medium containing what 3S35, sulfur 35, some other bacteriophage they cultivated in the medium. And actually, that radioactive phosphorus, that is P32, provide radioactivity only to the DNA present in the bacteriophage. You know, bacteriophage consists of only two things. One is DNA inside and the protein coat outside. So if current comes, the same problem will be there.
Yeah, when current comes also. There was one setting, I think.
Yeah. Okay, welcome back. Here, current has gone for a minute. Sorry for that. So we will go for this experiment conducted by the Hershey and Chase. So as I told you, they kept this bacteriophage or the phage virus in the two media, and one media containing the radioactive phosphorus P32 and another media containing the radioactive sulfur ES35, 35S. Right. So actually, that the phosphorus present only in the DNA, maybe remembering the structure of the DNA, phosphorus is there. So whenever they are putting this bacteriophage in the radioactive phosphorus, that P32, naturally it provides the radioactivity to the DNA only, not not that protein. Whereas whenever they put this bacteriophage in the radioactive sulfur, that is S35, naturally radioactivity will be given to the protein coat of this bacteriophage. So they made the two type of bacteriophage here. One, they but you can say provide the radioactivity to the DNA molecule and also provide the radioactivity to the sulfur coat, and both the phage labeled with the phosphorus and sulfur allowed to infect the bacteriophage, or that is what E. coli or the bacteria. This both the phage. In one bacteriophage, DNA is labeled with the radioactive material, and another phage virus, that DNA, that protein only labeled with the radioactive material. So come to the different steps in the experiment conducted by the Hershey and Chase. One is infection. As I told you, simply make this bacteriophage to infect the bacteria. Then second one is what? After infection, what happened? Normally, bacteria send only the DNA molecule inside, but protein coat will be outside. And this protein coat can be removed viral protein coat can be removed from the surface of the bacteria through agitating in a blender. And it is called as what? The blending. Removal of the protein coat. After infection, the protein will be there outside, and DNA only it is sending inside, and this protein can be removed through what you can say agitation in a blender. We call it as what? The blending. Then afterwards we have to go for centrifugation. That is what? The separation of viral particle from the bacteria by spinning them using centrifuge. By using the centrifuge, that viral particle present in the bacteria can be separated, and it is called as what? The centrifugation. So there are three steps in the process of this experiment conducted by the Hershey and Chase: infection, blending, and centrifugation. And see this three steps in detail here. This is what? The infection. Bacteriophage infect the bacteria. And here two type of bacteriophage is used. One is what? Bacteriophage having what? Radioactive protein coat. And another one, the genetic material, that is DNA, is also labeled with radioactive phosphorus molecule. That is what? The infection. Infect the bacteria with this bacteriophage. And here, second one is what? The blending process. See, after sending that DNA into the bacteria, the protein coat will be there outside of this bacteria because it is not taken inside at the time of infection. And this protein coat can be removed by using what? Some agitator in the blender, and that process only we call it as what? The blending process. This process is called as what? The blending. By using that blender or agitator, it can be what you can say separated this protein coat from the surface of the bacteria, and this process only we call it as what? The blending. Then last step is what? The centrifugation. Maybe done the centrifugation process. By using the centrifuge, we can separate what? The DNA present in the what you can say bacteria sent by the phage virus. Separation of genetic content DNA of the phage virus from the bacterial body is called as what? The centrifugation. Right. Let us see what the result of the experiment. So bacteria which was infected with the bacteriophage labeled with the radioisotope P32. That means here they label only the genetic material DNA because phosphorus present only in the DNA, and they found the radioactive DNA. After extracting this DNA from the bacteria, they found the radioactive DNA here. And it indicates that DNA was the genetic material that passed from the phage virus to bacteria at the time of infection. That is why they found that the radioactive DNA inside the bacteria. After this centrifugation, they got what? The radioactive DNA from the body of this bacteria, because at the time of infection, what is happened? The DNA only sending inside, and they never show the radioactive protein inside. Why? Protein is not taken inside. So the protein coat produced by the bacteriophage in the bacteria never show the radioactive protein. Why? Because at the time of infection, it is taking inside only its DNA and not the protein outside. And by using that DNA taken inside the body of the bacteria, only it multiplies its number inside this host bacteria. And it is indicating that at the time of infection, radioactive protein not enter into the bacterial cell, only this DNA only taken inside. And this experiment help us to understand that the real genetic material taken into the bacteria from the bacteriophage is only the DNA, and by using the DNA only, it multiply its number in the host, that is bacteria, right? And this is what the experiment proved, that the real genetic material is DNA. So see the diagram, it's giving clear idea about this process: infection, blending, and centrifugation. And this is what the bacteria. You can see that red colored label here. That is what the sulfur labeled protein capsule only. The DNA is not labeled here with the radioisotope. Only protein is labeled by using the radioactive sulfur. And another bacteriophage that by using that phosphorus only it is labeled, radioactive phosphorus only it's labeled. So only DNA is labeled, but not the protein code. And allow these two bacteriophage infect this bacteria. See, after infecting this bacteria with this bacteriophage, it is sending only the DNA inside. You can see that the black colored DNA only is taking inside, and it is not radioactive. But the radioactive phosphor, radioactive sulfur present in this protein code remains outside only, and that will be outside. And through the process of blending, it can be separated, and it go for centrifugation and collect what the DNA from the body of the bacteria, and they never find the radioactivity in any bacteriophage multiplied inside the body of bacteria. Many bacteriophages multiplied inside the bacteria, but they never find the radioactivity in any kinds of bacteriophage multiplied inside the body of bacteria. That means the protein is not having any role here for its multiplication, because it is not the real genetic material, but they detected this what the radioactive sulfur here from the separated that protein capsule. And come to the other side, they use what another bacteriophage where the DNA only labeled with radioactive sulfur, radioactive phosphorus, and allow this bacteria infect with this bacteriophage. It is taking the DNA content only inside, and it is radioactive. And this DNA will go for multiplication, and finally produce what? The protein code. And finally, it after the blending and centrifugation, they got a result. This phosphorus detected inside the cell, because the bacteriophage produce inside the bacteria containing the radioactivity in their genetic material. That means DNA only taken inside the bacteria, not the protein. This is what the experimental proof for explaining the real genetic material is DNA is not a protein code, right? Then we will move to another same thing only mentioned here. This is what the phage virus infect the bacteria, and the protein is labeled here. It is given in red color, and it is what the process of what the blending, separating this protein code from outside, and after centrifugation, sulfur they didn't find inside the cell. And in second experiment, the DNA only labeled here with the phosphorus, the radioactive phosphorus, and it is taken inside and multiply, and because of the blending, this will be separated. And after centrifugation, they found the phosphorus inside the cell, that is radioactive phosphorus inside the cell. That means this DNA only taking inside the bacteria for the multiplication of bacteriophage, because the real genetic material is what? It's DNA only, not this protein. This is what the another what you can say diagram given: infection, blending, centrifugation, and this is what the observation. So S35 use here to label this the protein, and this is what the infection, and it is what the blending, removal of the protein code. After centrifugation, what you can say that S35 labeled phage protein found in the supernatant. There we can find what the protein taken from the cot. And another experiment here, they use what the bacteriophage label its DNA with P35 and allowed to infect this bacteria, taking this DNA inside and multiply it and go for what the blending, then go for the centrifugation. And after centrifugation, they found what is the S35 labeled phage protein found inside the cell. Right. And this experiment conducted by Hershey and Chase proved that the real genetic material is not a protein, and it is what the DNA only, because protein is not at all taken inside at the time of infection. It is only for the protection. It is not at all determine the characteristic feature of the individual, that is determined by the genetic material, that is DNA only. And here they have a protein code, and it is only for its protection. Then come to the properties of genetic material. Already we discussed some of the properties of the genetic material. See, genetic material should be able to generate its replica. Replica means what? Making its copies without any change in the chemical nature. And this process is called as what? Replication, DNA replication. We will see it later in detail. See, after taking that DNA from the bacteriophage into the bacteria, and this DNA of this bacteriophage start multiplying its number by using all the machinery present in the bacteria. It is what? Replicating. Replicating means making many copies without making any kinds of changes in their genetic makeup. And it should be chemically and structurally stable. That the genetic material should be stable chemically and structurally. And it should provide a scope for slow changes through mutation that are required for evolution, maybe remembering this point that we discussed in the morning. And it should be able to express its character in the form of what? Mendelian characters. It should be able to express, because genetic material should store the information about a particular character, and whenever it required, it should express the character in the form of what? The Mendelian characters only. And DNA only fulfill all these characters, not protein or not RNA. That is why nature select DNA as what? The genetic material for all the individual except the retroviruses. Right? Then DNA and RNA are able to replicate. Protein cannot multiply. Then genetic material should be resistant to changes with the different stages of life cycle. It should not deviate according to the age, life cycle, or physiological aspects of the individual. That should resistance to what you can say changes with different stages of life cycle, age, or change in the physiological aspects of the individual. And see, the Griffith proved in transforming principle is that heat killed genetic material regain its character when it gets what? The appropriate condition. See, whenever we kill the bacteria by using the heat, it what you can say lose its size. But whenever it get the opportunity, whenever it get appropriate condition, what happened? It became very, very functional. That is why it convert this R strain into what? The virulent S strain. Genetic material should be resistant to changes. And double-stranded DNA can be separated by heating. I told you what you can say, hydrogen bond is there between the double strand structure of the DNA. That can be separated, can break by using what? The heat energy. But whenever this what you can say separated DNA strand get opportunity, get the suitable condition and temperature, again it get what? The double-stranded structure back. Double-stranded DNA can be separated by heating, but it can get back its double-stranded structure after providing an appropriate condition. That means it is somewhat stable, and it is resistant to the changes with the different what you can say stages of life cycle, age, or with the change in the physiological aspect of the individual. See, more than that, DNA is more stable than RNA due to several other reasons. In morning, we have discussed some of the reason for making this DNA more stable than RNA. So DNA is what? The better material acting in the body of an individual as a genetic material than what you can say this RNA. Then come to RNA world. See some things only mentioned here about this RNA and DNA. As I told you in the morning, RNA was the first genetic material. Initially, RNA only originated. Essential life process such as metabolism, translation, splicing, etc. evolved around RNA only. But many biochemical reactions in the living cells are also catalyzed by the RNA only, not by what you can say protein enzymes. As a catalyst, RNA is very reactive and hence unstable. Now we are discussing why RNA cannot act as for the genetic material, efficient genetic material, because it is not that much stable, because it is what working in the cell as a catalyst, and it catalyzes many type of biochemical reactions in the living cell, and therefore DNA has evolved because of what the less resistance to the changes. This DNA has evolved from the RNA with the chemical modification that make it more stable, and DNA being double stranded and having a complementary strand further resist changes by evolving the process of repair. So it is double stranded. Something happened to one strand that can be repaired by using what the another strand. In case of RNA, it is not there. RNA is single stranded. Something happened chemically. It is not possible. No scope for any kinds of repair, because DNA is double stranded and both the strands are complementary to each other. Something happened to one strand in its chemical nature, and it can be repaired by seeing what the complementary strand present here, because it is what the double stranded only. That scope is also there for the DNA, and because of all these things only, DNA is acting as what the genetic material. So we will move to another important topic here, and that is what the DNA replication. Just now I told you about one of the most important characteristic feature of the genetic material, that is what the replication or the multiplication only. Replication is what the process to multiply the content of the DNA without making any change in its chemical nature, and that only referred as what the DNA replication. So DNA replication is the process of multiplication of DNA content during the S phase in mitosis. You may be heard about the different phases in the interphase of this mitosis, like G0 phase, G1 phase, S phase, G2 phase, etc. And there is no production of new DNA during the G1 phase or G2 phase. Only what the growth take place due to the synthesis of what protein, lipids, etc. and other biomolecule. But S phase, it is what the synthetic phase. Why it is called as synthetic phase? Because in this phase only the total DNA content present in the cell multiply, and it synthesize the DNA through the process of what the DNA replication only. And DNA replication is essential for doubling the chromosome number for the equational division. I told you yesterday, total number of chromosome in the human cell is 23 pairs only. And whenever this cell undergo the division, mitotic division, and before this cell division or cytoplasmic division or nuclear division, the total content of the DNA present in the nucleus will multiply, or chromosome number will multiply. Then only when only the nucleus can go for division and produce two equal nuclei. Right. That process only we call it as what the DNA replication. DNA replication is essential for doubling the chromosome number for the equational division, and DNA replication is semi-conservative. Remember the term semi-conservative, because during this DNA replication, one strand is conserved and another one is newly constructed. That is why we call it as what semi-conservative, not fully conservative. Fully conservative means what both the strands are conserved, but here only one strand is conserved and another one is newly constructed. That is why we call it as what semi-discontinuous, or what the semi-conservative method of DNA replication. And this process only we are going to discuss here in very detail. And this topic is very important as far as your examination is concerned. This DNA replication asked many times in the board examination. So we have to learn in very detail the process of DNA replication. So process of DNA replication is nothing but multiplying the total content of the DNA without making any changes in the chemical nature of the DNA. It is making exact Xerox copy of the DNA, and every DNA, every chromosome present in the nucleus of the cell undergo the DNA replication to duplicate the chromosome just before the what you can say mitosis, that is what the equational division. And see, total length of mammalian DNA is roughly around 2.2 meters. And it consists of 6.6 into 10 raised to 9 base pairs in the total length of the DNA in a mammalian cell. The speed of replication, increasing the speed of replication is about 2000 base pair per second or 100 nucleosome per second. This is what the speed of what the replication, making a new strand. That is what 2000 base pair per second or 100 nucleosome per second. Then DNA replication essential for chromosomal duplication before mitosis. Just now I told you, through the DNA replication only we will able to duplicate the chromosome. 23 pairs of chromosome we have. Before this nuclear division, it should be doubled through the chromosomal duplication, and exactly it duplicate the chromosome only through what the DNA replication, because the main content of the chromosome is DNA only. Told you in the morning, and DNA should replicate without making any changes in the chemical nature. Then only chromosome can duplicate before this nuclear division during the mitosis. And this help for the equational division and also help to maintain total number of chromosome constant. See, take any cell present in our body, all the cells of our body is derived from the zygote only. Zygote is what the product of what the fusion of male and female gamete having 23 pairs of chromosome. Whenever these cells divide repeatedly through mitosis, many cells will be formed. All cells are equal in its what the chromosome number, and this cell only present in our body after the completion of growth. So naturally, every cell you can take from the body from anywhere, we can find the same number of chromosome, 23 pairs of chromosome only, and it is constant all the time. And it's happened during the S phase of the interphase in meiosis. So this chromosomal duplication, doubling of chromosome is happening during the S phase of the interphase of the mitosis, and chromosomal duplication is possible only through what the multiplication of the DNA through this DNA replication process. And see the role of enzyme for the DNA replication. We will see first the enzymes helping for the DNA replication, and also we'll discuss some technical terms. See, after discussing in detail about this, what the enzymes and technical term, we will go directly into the real process of DNA replication. And see the role of some enzyme for DNA replication. You have to note down all these enzymes and its function. Whenever we discuss the process of DNA replication, we will find where its work. This helicase enzyme, it's what enzyme, very important enzyme for unwinding of double-stranded DNA by breaking the hydrogen bond. You know, DNA is what the double-stranded structure. It is antiparallel, and this antiparallel double-stranded DNA is connected each other with a hydrogen bond and form what the nitrogenous base pairs. And to separate these two strands during this DNA replication required helicase enzymes to cut the what you can say this hydrogen bond and making these two strands separate each other. Here you can see that diagram here. You can see the diagram here. Right here, this is what the DNA strand, double stranded, and it cut its hydrogen bond and separate. That is called as what the unwinding of the DNA strand. And this unwinding of the DNA strand required these enzymes. enzymes. And the name of the enzyme responsible for unwinding of the double strand by breaking the hydrogen bond is called as what? The helicase enzyme. Then topoisomers. It is to reduce the tension while unwinding. You know, it is what you can say double stranded. DNA is what? The double stranded. Go for coiling in right hand position. After breaking the hydrogen bond, these two strands start separate each other, unwinding. That create lot of tension in another area. And to reduce the tension while unwinding, this we are using what? The topoisomerase enzyme during what? The DNA replication. Topoisomerase enzyme help to reduce the tension during the unwinding of the double-stranded DNA after breaking the hydrogen bond with the help of the helicase enzyme. Then another one, that is what? The RNA primers. And we will see that RNA primer while discussing about this replication process. And it is produced by what? An enzyme called as what? The RNA primase only. RNA primer is a small RNA nucleotide only. And this is acting as what? The basement. And it is helping for attaching the DNA polymerase to extent. So while discussing about the replication, we will see the role of what? The RNA primase enzyme. Then DNA polymerase. DNA polymerase only elongate the new DNA strand. I told you it is what? The semi-conservative one only. One strand is already there, and they are making what? Another strand by considering what? This strand. We call it as what? The master strand, right? And this elongation of the new strand by using that old strand with the help of the enzyme called as what? The DNA polymerase enzyme. And it is to elongate the DNA strand from 5' direction to 3' direction. Always you remember this enzyme, DNA polymerase, and we will see RNA polymerase also later during the process of transcription. All this polymerase enzyme is a DNA or RNA polymerase, always elongate the new strand always from the 5' direction to 3' direction only. And it is not only for elongation, the DNA polymerase, it is also used for the proofreading. See, after constructing a new strand, chances are there for some kinds of mistake somewhere. And this mistake can be found out and it can be corrected or it can be repaired by using what? The same DNA polymerase only. And this process only we call it as what? The proofreading. And DNA ligase, it is to join the Okazaki fragments. We will see what is Okazaki fragments. Maybe learn the Okazaki fragment. These are joined together to form a continuous DNA strand with the help of what? The ligase enzyme. It is acting as what? The glue to bind the what you can say fragments of the DNA. And hexokinase enzyme, that is to remove the RNA primer. After constructing a new strand of DNA, that RNA primer should be removed, and that is with the help of what? The hexonuclease enzyme only. So these are the 6 enzymes responsible for this DNA replication. They have their own special role during the process of DNA replication. Helicase enzyme, then topoisomerase, then RNA primase, then DNA polymerase enzyme, then DNA ligase. The last one is what? The hexonuclease enzyme. So we will see a role of all the enzyme later while discussing about the real process of what? The DNA replication, right? Then see the another one, the terms. So first we will see the terms, some terms we will discuss while we will see some kinds of terms while discussing about the process of DNA replication. Yeah, you can also check while discussing about this replication process. One is what? The Ori site. You may be heard about the site, Ori site. This is what the site from where the replication originates. The real process of replication originates from these sites. We call it as what? The Ori site. Then another one is what? The lagging strand. One strand, new strand produced in the form of what? The small strips of DNA. And because you know that DNA polymerase enzyme elongate new strand only from 5' direction to 3' direction. So chances are there to produce, because of that, chances are there to produce new DNA strand in one side in the form of small fragment. And it is only, we call it as what, the Okazaki fragment. And that strand with this Okazaki fragment only, we call it as what, the lagging strand. Whereas another side, you know, it will move continuously from 5' to 3' direction, and we call it as what, the leading strand only. So lagging strand, leading strand, then RNA primer, a small RNA polypeptide only fixed there for attaching this, what, the enzyme called as what, the DNA polymerase enzyme, and Okazaki fragments, then proofreading and DNA repair. Proofreading means what? Making changes when it is mistaken somewhere while making the new strand. That is proofreading by our DNA polymerase enzyme only. Then semi-conservative method of replication. And one strand is conserved during the process of replication. That is why we call it as what, the semi-conservative. Then continuous strand and discontinuous strand. One strand is continuous, another one is discontinuous. Then template or master strand. After separating the DNA, both the strands are working as what, the master strand or template. Then replication fork. After winding, it get what, fork-shaped structure. And unwinding of DNA. Unwinding of DNA, you know, it is in the coiled form only, winding form only. And after breaking the hydrogen bond, it go for unwinding. So these are some of the terms related to the DNA replication, right? And see the process of DNA replication in detail. So where the DNA replication take place? In a cell. You know, DNA is the most important part of the chromosomes only. And this chromosome is located in the nucleus. So naturally, the replication of the DNA also take place in the nucleus. Because of the replication of the nucleus in the replication of the DNA in the nucleus, that chromosome content will increase. So it become, that nucleus become very spherical just before the nuclear division. The process of DNA replication start from different part of the DNA. You know, DNA is very elongated, and it is not possible to replicate the entire length of DNA to start from one end to another end. It's take a lot of time to complete the replication of the entire length of the DNA. That is why this DNA replication start from different part of the DNA, and it is not possible to replicate the entire length of DNA from one end to another end within a stipulated time. So E. coli like bacteria take only less than 20 minutes to multiply the total number of chromosomes, because it is so quick and very speed. The place from where the process of replication start, it may be from one end or it may be in between. And this portion of where this process of replication start in DNA is called as what, the ori site. We mentioned while discussing about the term ori site, or we call it as what, the origin of replication. And replication of DNA take place by using different ori site in the same time in DNA at the same time, in same DNA at the same time, because it is not possible to start from one place. Required many places to start the replication to finish it so easily by taking only less time. So many ori site will be there in a DNA at the time of what, the DNA replication. What is ori site or the origin of replication? This is what the place in a DNA from where the real process of what, the DNA replication originate, right? And see the process here. Here, this is what the DNA molecule, double-stranded one only DNA molecule. It can be start from the end, or it can be started from some other areas of the DNA. If it is from the end port, we will get a fork. We'll see the fork. And if it is from here, we'll get what, a bubble here. Origin of replication, we'll get a bubble. It is what, the DNA strand. And it can be start from here. If it is start from here, it make the fork. And if you are the DNA is starting it from the center, you will get what you can say a bubble shaped a bulged portion, and it is called as what the bubble, origin of replication only, right? And here come to the unwinding of the DNA. Which enzyme is helping for breaking the hydrogen bond and to separate the double-stranded DNA? That is helicase enzymes only. We discussed about the role of this enzyme, helicase enzyme. And this enzyme helps to break the hydrogen bond between two DNA strands. And after breaking the hydrogen bond, these two DNA strands start to move apart each other, and that only we call it as what the unwinding of the DNA. Unwinding of DNA. Here you can see what the separation of the DNA. Separation of the DNA after breaking what this hydrogen bond. Hydrogen bond only connect one strand to another strand. These strands are anti-parallel, 5' 3'. Another one is 3' 5'. And here these two strands separated with the help of what helicase enzyme that break the hydrogen bond. It start here and it move in this direction. While moving, it break all the hydrogen bond. And after separating these two strands, after breaking the hydrogen bond, it starts separate each other, repel each other, and get what a fork here. And it is called as what the replication fork. This is called as what the replication fork. Then see another one. Here we can see another helicase enzyme here. This is what the helicase enzyme. It break the hydrogen bond. This is what the DNA strands. DNA strands only, and this is what the origin of replication. And replication start here. And this helicase enzyme break the hydrogen bond and start separating these two what you can say complementary strands of the DNA, and get what you can say a bubble here called as what the replication bubble. I told you about the replication fork and the replication bubble. It is due to the separation of what the complementary strands of the DNA after breaking this hydrogen bond only, right? And here come to the unwinding of the DNA. After breaking the hydrogen bond by using the helicase enzyme, these two strands of the DNA, complementary strands only, move apart each other, and it's called as what unwinding of DNA. Then it results the formation of a replication fork or that bubble, replication bubble. If it is in between, it produces what the bubble, replication bubbles. Due to the separation of these two strands of DNA, create a lot of pressure in the other area. Whenever it's separate, lot of pressure will be created in another area that can be reduced by another enzyme called as what the topoisomers. We'll show you one slide here. You can find the separation of what the DNA strand, and this is what the double-stranded DNA, and this is what the site of the ORI site where the process of replication originates. And the helicase enzyme only break all the hydrogen bond. And after breaking the hydrogen bond, naturally these two strands separate each other. And this repulsion of these two strands create a lot of pressure here, lot of tension here. And this can be reduced by using what topoisomers enzymes only. So helicase enzyme to break the hydrogen bond and also helping for the unwinding of the DNA strand. Whereas the topoisomers enzyme reduce tension produced here in the another end due to the unwinding or separation of these two complementary DNA strand of DNA. Complementary strand of this DNA. So helicase enzyme to break the hydrogen bond and topoisomerase enzyme to reduce the tension in this region during what the separation of complementary strand of DNA, right? And see another one. After unwinding of DNA by breaking the hydrogen bond, both the strands of DNA, there are two strands of DNA. After unwinding this DNA, double-stranded DNA. One is what the 3' 5' strand, another one is what the 5' 3' strand. And these two strands are acting as what the template strand. While discussing about the term, I told you about the template strand or the master strand, and both act as a template strand or the master strand. Master strand or template strand means what? The new DNA is synthesized according to this separated Vatican said DNA strand only. And such type of strand only we call it as what? The master strand. See, after separating the hydrogen bond, these two strands of the DNA repel each other and go for unwinding and creates a lot of pressure here. Topoisomers only Vaticans reduce the pressure formed here, and helicase enzyme only break the hydrogen bond between the two strands of DNA. And after unwinding of the DNA, these two strands, one is 5' 3' strand, another one is 3' 5' strand. Both the strands act as what? The template or the master strand only. There are two templates here, and one template is running from 3' 5' direction. Another template is, anti-parallel only, that is running from 5' 3' direction. Both the strand of the DNA is acting as what? The template only. And based on this template only, new strand is constructed. As I told you, it is what? Semi-conservative. And during this, what you can say, DNA replication, one strand is conserved and another one is newly constructed. So here, new strand is constructed according to the nitrogenous base sequence present in the template or the master strand only. And this master strand only give information what type of nitrogenous base should be placed for the new strand. And here also, new strand will be constructed based on the nitrogenous base present in this old strand only. That is why we call it as what? The template strand or this master strand. New DNA strand is constructed because it is semi-conservative only. And whenever it is constructing a new strand here, this sequence of nitrogenous base should be based on what? The master strand or the template strand only. Right. So after unwinding DNA by breaking the hydrogen bond, both the strands of the DNA act as a template or the master strand on the basis of deoxy polynucleotide present in the template or the master strand. Suppose it containing A or containing T or it's containing what? The G or C. That is what the deoxy polynucleotide. Remember the things that we learned earlier. On the basis of such a polynucleotide present in the template strand, DNA polymerase enzyme plays its complementary strand in the new strand of nucleotide in the direction 5' 3' direction, and it is taken from the nucleoplasm only. See, all type of deoxy polynucleotides are there. Deoxynucleotides are there in the nucleoplasm, and according to the what you can say nucleotide present in the template, this DNA polymerase will place all these corresponding deoxynucleotide in the new strand in the direction, and it's elongate from 5' 3' direction only. But formation of new strand of DNA required a basement that is called as what? The RNA primer, and it is produced by the RNA primase only. It is not possible to elongate the new strand of DNA, and that the DNA polymerase should bind to the RNA primer, then only it can elongate in the direction 5' 3' direction only. So RNA primer is nothing but a small RNA nucleotides only, and from these RNA nucleotides only DNA polymerase elongate from the 5' direction to the 3' direction. So this RNA primer is produced here for the attachment of the DNA polymerase enzyme for its elongation with the help of the enzyme called as what? The RNA primase enzyme only. Like here we can see the template master strand. Two master strands are there. One is what? 3' 5' strand, another one is 5' 3' strand only. And after separation or unwinding, both the strands of the DNA is acting as what? The template only. Then the DNA strand as a template having 3' 5' direction produce a continuous complementary strand. Why this DNA strand, the template strand having 3' 5' direction produce a complementary and continuous strand? We call it as leading strand because new strand of DNA always elongate 5' to 3' only. So the master strand having 3' 5' direction produce what the corresponding strand with the help of the DNA polymerase continuously. Only one primer is required here for attaching the DNA, and from this primer it go for elongation from 5' direction to 3' direction continuously, and that only making what the leading strand. Here you can see that the leading strand here. This is what the separated all the DNA strand, and these two strand only acting as what the template. And this template is having 5' 3' direction, and another one is what the 3' 5' direction only. According to the nitrogenous base pair present in the template strand, RNA polymerase will place what the nucleotide one after another taken from the nucleoplasm, but this DNA polymerase enzyme always elongate the DNA strand from 5' direction to 3' direction only. So the master strand having 3' direction here. First they produce what a primer here, and DNA polymerase attached here. So DNA polymerase, after attaching the primer, it can be taken continuously because elongation is always from the 5' direction to 3' direction only. And this is what the new strand. It is what the old strand. Old strand 5' 3' 5' direction. So naturally old strand should be complementary and anti-parallel, and it should be running from 5' direction to 3' direction. So here DNA polymerase always permit the elongation of the new strand always from the 5' to 3' direction. But to attach the DNA polymerase enzyme required a primer. It is RNA primer only. It is a small RNA molecules only, and from this RNA molecule only the DNA polymerase start its elongation from 5' direction to 3'. So this strand, the new strand constructed by this, what you can say, this master strand having 3' 5' direction always produce a continuous strand. And here this strand is called as what the leading strand. This DNA strand is called as what the leading strand because it is not making the DNA strand step by step. It is what the continuous only, and we call it as what the leading strand. And in another side, this is what the all the DNA strand running from 5' direction to 3' direction only. So the DNA polymerase never elongate from the 3' to 5' direction. So it should be complementary and anti-parallel. So it should be 3' here. It is not possible to elongate a new DNA strand from the 3' to the 5' direction. So what happened? It is making a small primer here, and this from this primer it is elongating new DNA strand from 5' to 3' direction, and another primer will be placed here, and from this what RNA primer new strands will be elongated from the 5' to 3' direction. So like that, from 5' to 3' direction only this DNA polymerase elongate the new strand of DNA. So it is not possible to produce a continuous strand here in this what you can say side, because here master strand is what the 5' to 3' only. So the newly constructed strand should be from 3' 5'. So it is produced in this side in the form of what the discontinuous strand only. Here we can find so many fragments of the DNA, and these fragments only we call it as what the Okazaki fragments. In one side, this new DNA is constructed continuously, whereas another side, DNA molecule is constructed discontinuously, bit by bit or fragment by fragment. And these fragments are called as what the Okazaki fragment. Why it is producing in the form of Okazaki fragment? Because always DNA Polymerase enzyme elongate a new DNA strand from the 5' direction to 3' direction only. Always you have to remember elongation of the new DNA strand, formation of new DNA strand always from the 5' direction to 3' direction. So this side only one primer is required. Only one primer is required to elongate a new strand here, whereas this side for constructing every Okazaki fragment required this RNA primers, because RNA primer is for attaching the DNA polymerase enzyme, and this DNA polymerase enzyme plays what this deoxynucleotide one after another according to the nucleotide present in the master strand. Suppose in master strand A is there, so naturally that nucleotide having T only plays here. If it is G here, naturally C only plays here. So like that, it should be complementary. The respective nucleotide having that particular nitrogenous base, it is collecting from the nucleoplasm. Nucleoplasm is having all kinds of nucleotide. That particular nucleotide will be collected by this DNA polymerase enzyme, and it attach one after another and go for elongation, but always from 5' direction to 3' I mean from 5' direction to 3' direction only. That is why it is producing in the form of small fragment in this side, and it is called as what? The lagging strand. This is what the continuous one, and we call it as what? The leading strand. These are the some of the term we mentioned earlier. DNA and what you can say, unwinding here, and zip DNA, separate the DNA fragment, continuous strand, then discontinuous strand, Okazaki fragment, then that leading strand and the lagging strand. So these are the some of the technical term mentioned in the textbook. Ori site we already discussed. This is what the site from where this the real process of what the replication originate or start, and after starting the what you can say replication, first it to go for breaking the hydrogen bond, unbind it, and produce what the replication fork, and that will create a lot of helicase enzyme only helping to break the hydrogen bond, and topoisomers reduce the tension formed here due to the unwinding, and a new strand formed continuously in that template strand running 3' 5' direction, and it is called as what? The leading strand only, and the new strand constructed here is continuous, called as what? The continuous strand, because only one primer is fixed here in this 5' end, and from where DNA polymerase elongate new strand of DNA continuously. Whereas another side, the template is what? The 5' 3' only, and it is not possible to extend the DNA from 3' to 5'. DNA polymerase always extend the new DNA in from the 5' direction to 3' direction only. So it is produced in this side, the new DNA in the form of what? These small fragment. So for making every fragment required the RNA polymer. Every fragment required RNA polymer. From there only DNA polymerase enzyme extended from 5' to 3' direction. So this is what the direction of producing new strand of DNA in this side. And here the new DNAs are produced discontinuously, and in the form of fragments called as what? The Okazaki fragment, and this strand is called as what? The lagging strand. Lagging strand, and this one is what? The leading strand. It is a newly constructed continuous strand, and it is newly constructed discontinuous strand. We call it as what? The Okazaki fragments, right? Then see this construction of new DNA. See, actually, this DNA polymerase enzyme only to elongate the new DNA. And this elongation of the DNA polymerase enzyme is from 5' direction to 3' direction only all the time. All the time it elongates a new DNA strand from the direction 5' to 3' direction only. And see this template, it is 3' and it is 5' direction. And so naturally newly constructed DNA should be complementary and anti-parallel. So it is possible to start from the 5 prime and it is leading to what you can say 3 prime only. So it is possible to construct a new strand here based on what the nitrogenous base sequence present in the template strand can arrange what you can say according to the nitrogenous base of the template strand. Suppose here it is A, naturally here it will be T. If it is G, here it will be C, and it is C, here it will be G. If it is A, here it will be T, like that. One by one, all the nitrogenous base will be arranged according to the nitrogenous base present in the template, and DNA polymerase only elongate, but elongation always from the 5 prime direction to 3 prime direction. This is what new strand. Why it is called as semi-conservative? Because during the replication, what happened, that old strand is conserved, and this is what the newly constructed one. One strand is conserved and another one is constructed newly. And new strand is always elongate from the 5 prime to 3 prime direction only. And this is what the DNA polymerase, it is running in the same direction. Only one primer is required here. So from this primer, it can elongate from 5 prime direction to 3 prime direction. But it is not possible in the next side, because here master strand is what 5 prime to 3 prime only. So it is not possible to run from this end to another end, because replication, that unwinding is going on continuously here by breaking the hydrogen bond. And it is not possible to elongate from here to here, because DNA polymerase always elongate from the 5 prime to 3 prime direction only. If we start from here, 5 prime, 3 prime, if it goes like this, it should not be anti-parallel. This is what anti-parallel only. It is 5 prime, 3 prime. Here it is 3 prime, 5 prime. But if it is moved from here, it is not anti-parallel. To get the anti-parallel nature, it should be constructed from 5 prime to 3 prime direction like this. And this is what the what you can say primer here. From this primer only it's originate, and again primer should be placed here. RNA primer only. It is what a small length of RNA for attaching or binding this DNA polymerase enzymes only, and from there it's what elongate. And this is what another primer. From this primer it elongate, always elongation from 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime direction, and produce in the form of small fragment. That only we call it as what the Okazaki fragments, right? This is called as what the Okazaki fragments. See, after the formation of two new strands complementary to the old strand, and will be inspected to find out the mistake. That is called as what the proofreading. And here, after making the DNA here in the form of fragment, there is an enzyme called as what the ligase enzyme, call it as what the biological glue. What is the function of the glue? To connect it. And this ligase enzyme only connect all these small fragments of what the DNA called as Okazaki fragments together to form a continuous strand. Ligase enzyme, name of the enzyme to bind all these fragments together. It's called as what the ligase enzyme. But here ligase enzyme is not there required, because it is what the continuous strand only. It is a discontinuous strand produced in the form of what small fragment, and later this fragment should be bind together to form a continuous strand. That is done by the ligase enzyme only, right? Then after the formation of these two strands complementary to the old strand will be inspected to find out the mistake. That only we call it as what the proofreading. Proofreading. See, after constructing the DNA, new strand DNA strand complementary to what the template. This is what the new strand constructed complementary to the what you can say template. It should be inspected from one end to another, because chances are there for some kinds of mistakes. Suppose here it is A, chance is there to place what the C here, and it is what the mistake only. That mistake should be find out, because the DNA replication is producing what the exact copy, and it is what the replica, no change in the chemical nature. That should be exactly like what the parental DNA only. So it should require some kinds of mechanisms to find out the mistake, and that only we call it as what the proofreading. And proofreading is normally done by the DNA polymerase. Only it's having dual function. DNA polymerase is having dual function. The first function of the DNA polymerase is to elongate the new strand of DNA from the 5' direction to 3' direction only. One duty. And the next duty of the DNA polymerase enzyme is to inspect the entire length of the DNA to find out the mistake. And this process only we call it as what the proofreading. DNA polymerase go for the proofreading to identify the mistake. Inspect from one length to another length. After constructing that new strand of DNA, polymerase enzymes run from one end to another to inspect, to find out the mistake. And that only we call it as what the proofreading. And after proofreading, if any mistake they found, naturally they will replace and they will repair it. So after the formation of the two new strands complementary to the old strand will be inspected to find out the mistake. That only we call it as what the proofreading. And proofreading is also done by the DNA polymerase enzyme only. If it finds any kinds of mistake in the nitrogenous base sequence in the newly constructed DNA, will be corrected by the same DNA polymerase enzyme. So it is having the dual function. Once it is asked in the examination, the functions of DNA polymerase enzyme, not only for elongation of the new strand of DNA during DNA replication, it is also helping for the proofreading and also helping for the repairing. If they found any mistake anywhere during that inspection, it find it and replace and fix the correct one. That is also done by the DNA polymerase enzyme only, right? And here we will see the replication of DNA. We'll go through once again the DNA replication, and you will get a better idea, right? The process of what the DNA replication, it is very important. And we have discussed some what you can say the role of enzymes for the DNA replication. Go through it once again. Helicase enzyme divide the hydrogen bond between the double-stranded DNA and unwind, separate these two strands, that is helicase enzyme. While unwinding the double-stranded DNA, naturally tension will be there in another side that can be reduced by using what? Topoisomerase enzyme. Then what is the role of the RNA primers? It is to produce RNA primer. RNA primer is what? A small strand of RNA, and it is necessary to bind the DNA polymerase to elongate from 5' direction to what? The 3' direction. That is what the role of primer. It is synthesized by an enzyme called as what? The RNA primase enzyme. Then DNA polymerase enzyme, it is to elongate a new strand of DNA, as I told you, from only 5' direction to 3' direction, and also used for proofreading. After constructing it, inspect from one end to another end to find out the mistake, to correct it. And it is called as what? The proofreading and repair, that also done by the DNA polymerase enzyme only. And Okazaki fragment produced in the master strand having what? 5' 3' direction, that can be joined together to form a continuous strand with the help of the enzyme called as what? The biological glue or the ligase enzyme, DNA ligase. DNA molecule can be bind together or glued together, and the hexonuclease enzyme. See, after constructing all these Okazaki fragment, for construction for this Okazaki fragment required that RNA polymerase, RNA primer. After construction, that RNA primer should be removed. Then only this ligase enzyme can join it. So the removal of that RNA primer after the construction of the Okazaki fragment, even the continuous strand also start with the RNA primer only, but it should be removed after the completion of the replication, and that is done by what? The hexokinase enzyme only. I think you maybe knowing the role of different kinds of enzyme during the DNA replication, and see what the terms and terminology that we used during the process of DNA replication. Origin, it is what the place from where this replication originate. Then lagging strand, leading strand. One DNA strand after this, what the separation. Lagging strand is what the 5' 3' strand only. The lagging strand, leading strand is what the 3' 5' strand. RNA primer, it is what the small fragments of RNA from where this DNA polymerase elongate from 5' to 3' direction. Then Okazaki fragment, small fragment produced in the side. Their master strand is what the 5' 3' strand only. And proofreading and DNA repair, inspect the endial length of the newly constructed DNA by this DNA polymerase enzyme to find out the mistake. Then semi-conservative method of DNA replication. During the replication of the DNA, only one strand is conserved and another one is what the newly constructed, and one is continuous strand and another one is discontinuous strand. They are only producing what the Okazaki fragment. The template or master strand, that master strand or template is nothing but the old strand of DNA that only giving information regarding what the chemical nature of what the newly constructed DNA. Replication fork, after breaking the hydrogen bond, both the strands will be separated and produce a fork shape. That is called as what the replication fork. And unwinding of DNA, that means winded DNA, that coiled DNA, after breaking hydrogen bond, it starts separating each other, and we call it as what the unwinding only. Right? That's all about what the process of DNA replication. And we will move to another one, that is what the difference between the DNA replication in prokaryotes and eukaryote. We will see the differences in the evening, and all of you go for taking your teas. Now time is 4 o'clock, and we will meet at the 6:30 with this topic, the difference between the DNA replication happening in the body of prokaryotes as well as in the eukaryotes. So thank you so much. We will see in the evening. Thank you. Thank you so much.